All right, let's jump straight into this explainer. I want you to start by picturing a massive logistical nightmare. Imagine you're tasked with perfectly copying millions of incredibly complex data points, splitting them right down the middle, and moving them into two separate, totally identical facilities. You got to do all of this without losing a single line of code, and you have to pull it off in a matter of hours. Well, in the biological world, our bodies are solving this exact dizzying problem every single day. In fact, just since you clicked play a few seconds ago, millions of cells in your body have completely and perfectly divided to keep you alive and functioning. It's mind-blowing. And the elegant, beautifully choreographed biological solution to this massive data sorting problem, that's what we call mitosis.
To keep things perfectly clear, here is our roadmap for the explainer. 1. Introduction to mitosis. 2. Prophase, setting the stage. 3. Metaphase and anaphase. 4. Telophase, winding down. And finally, 5. Cytokinesis, the final cut.
Okay, let's kick things off with Part 1, Introduction to Mitosis.
So at its core, mitosis is fundamentally a process of nuclear division, and it occurs exclusively in our somatic cells. That's just a fancy way of saying our regular body cells. Think skin, liver, heart, and muscle tissue, as opposed to our reproductive cells. Now, the ultimate biological goal here is to produce two daughter cells, but the absolute most important keyword you have to remember is identical. Each new cell must contain the exact, unblemished, identical number of chromosomes as the parent cell. And why does this matter so intensely? It's all about tissue integrity. If your somatic cells didn't produce entirely identical copies during division, the structural and functional integrity of your body's tissues would start breaking down almost instantly. This flawless duplication is exactly how a skin cell replaces another skin cell with the precise genetic instructions it needs to keep doing its job.
Moving right along to Part 2, Prophase, setting the stage.
This is where our main characters, the chromosomes, step onto the stage and really begin their intensive prep for the performance. Notice how the loose, thread-like chromatin begins to shorten, thicken, and condense into those highly visible, dense structures we recognize as chromosomes. To really appreciate the scale of this logistics problem, remember that if you stretched out the DNA in just one of your microscopic cells, it would be about 2 meters long. Packing all of that into dense, movable units without tangling it up is an absolute marvel of biology. By the time they're visible, each chromosome actually consists of 2 identical copies called sister chromatids, and they're held tightly together right at their center by a structural belt called the centromere.
Let's break down all the moving parts here to ensure we don't miss any of the intricate choreography. While those chromosomes are busy condensing, the cell's centrosomes are physically moving away from each other toward opposite poles of the cell. You can kind of think of them like massive construction cranes setting up scaffolding on opposite sides of a build site. As they move, they extend microtubules which form the spindle fibers. These are basically the biological cables that will eventually move our cargo. And crucially, the cell is dismantling the vault. The nucleolus disappears, and the nuclear envelope, which is that highly protective wall normally surrounding your DNA, completely disintegrates, officially releasing those perfectly packed chromosomes right into the open cellular space.
Let's jump to the climax of our dance. Part 3, Metaphase and Anaphase.
What's genuinely fascinating right here is the sheer, undeniable precision of metaphase. The chromosomes perfectly line up right down the middle, at the equator of the cell, which we call the metaphase plate. Notice those spindle fibers extending from the centrosome cranes at the opposite poles? They reach out and attach directly to the centromeres of each individual chromosome. Imagine this as a super high-stakes, microscopic tug of war. The motor proteins are literally testing the tension, pulling back and forth. And here's the crazy part. The cell actually has built-in checkpoints and will absolutely refuse to proceed to the next step until every single chromosome is perfectly anchored and that tension is flawlessly balanced across the equator.
And then the tension breaks. In a sudden, violent snap, we enter anaphase. The centromeres split. Those sister chromatids, which by the way we now officially graduate to calling full daughter chromosomes, are rapidly pulled apart. This is the absolute shortest and most fast-paced phase of mitosis. The biological glue holding them together just dissolves in an instant, and motor proteins reel them in, dragging the separate chromosomes toward opposite poles of the cell along those microtubule highways. By the end of this incredibly fast sprint, both poles of the cell have secured an exactly equivalent, complete set of newly separated chromosomes. The logistical transfer of data is complete.
Now we take a breath with Part 4, Telophase, winding down. You can think of this phase as essentially going in exact reverse from prophase. Now that the cargo is successfully and safely arrived at opposite poles, it's time to unpack the boxes. The chromosomes begin to uncoil and lengthen, decondensing back into that loose, relaxed chromatin so the genetic instructions can actually be read and used again. The cell immediately gets to work rebuilding the vault. A brand new nuclear envelope forms around each complete, identical set of chromosomes at opposite ends of the cell. The nucleolus reappears, and those incredible spindle fibers that did all the heavy lifting, they completely disintegrate, having finished their job. So we now officially have two complete daughter nuclei residing securely inside a single cell.
Which brings us to part 5, cytokinesis, the final cut.
Because obviously having two nuclei stuck in one cell isn't the final objective of our logistical puzzle, right? So we reach the curtain drop known as cytokinesis, which is the actual physical division of the cytoplasm to form two entirely separate entities. In an animal cell, you'll see a shallow groove forming on the surface. This is called the cleavage furrow. What's fundamentally happening here is that a contractile ring made of actin and myosin microfilaments, fun fact, these are the exact same proteins responsible for your muscle contractions, is actively pinching the cell membrane. It acts just like a relentless mechanical drawstring, pulling tighter and tighter from the outside inwards, squeezing until the cell membrane is finally severed. And boom, it delivers two distinct, independent daughter cells.
But this mechanism reveals a really cool biological difference when we look at the broader tree of life. Plant cells cannot utilize a cleavage furrow. It really just comes down to structural engineering. Plants don't have skeletons. They rely on incredibly rigid cell walls made of tough cellulose to stand upright. And you simply cannot pinch a rigid cellulose wall in half with a microscopic drawstring. No way. So plants solve this logistical hurdle from the inside out. During telophase, vesicles, acting like tiny biological bricklayers, gather right in the center of the plant cell. They fuse together, physically depositing cell wall materials to build a brand new structure called a cell plate. This wall is constructed from the center outwards until it perfectly fuses with the existing outer edges, cleanly dividing the cell into two robust, walled compartments.
So as we wrap up this flawless, choreographed dance of identical division, it leaves us with a pretty provocative final question regarding the ultimate stakes of cellular control. The cell cycle, as we've seen, is strictly regulated by precise checkpoints, like when we were testing the tension on those spindle fibers. But what happens if that control is lost? Well, the biological reality is that a loss of cell cycle control at these crucial checkpoints can lead to abnormal runaway cell division. This loss of the brakes is actually the fundamental mechanism behind cancer and tumors. This beautifully complex, highly regulated dance is quite literally what keeps us alive and healthy, but it demands absolute, unforgiving precision at every single step. Thank you so much for joining me for this explainer, and keep questioning how the brilliant mechanics of the microscopic world perfectly shape our macroscopic reality.